up to the formation of peptides. Okay. The biological significance of your peptides. So number one, there are the so-called peptide hormones. So like your oxytocin and vasopressin. So oxytocin or vasopressin are cyclic nonapeptide. So when we say nonapeptide, they comprise of 9 amino acids joined together by your peptide bond. And they have identical sequences of amino acid except for two. Okay? So you have cysteine, asparagine, glutamine, isoleucine, tyrosine, cysteine. So cysteine and cysteine are within the vicinity of the first and the last amino acid of your peptide chain. That is the reason why they form a disulfide linkage. So same with your other amino acid. The only difference is that on the fourth amino acid, your oxytocin has isoleucine, while vasopressin have phenylalanine. Okay? Attached to your cysteine are your proline, leucine, and glycine. Okay? Again, if the N-terminal is glycine, you have arginine as the adjacent amino acid. In oxytocin, you have leucine. So only in isoleucine and leucine that they differ with phenylalanine and arginine. Having the same number of amino acids and differ only with these two amino acid, isoleucine instead of phenylalanine, leucine instead of arginine, they have differences in their function. So number one, oxytocin stimulates the contraction of uterine muscles and signals for milk production. This is the hormone that stimulates the uh, production of milk or during lactation. This is also, this peptide hormone also stimulate the contraction of the uterine muscles during the terminal stage of pregnancy, during labor or to induce labor. So those who pregnant women have high concentration of oxytocin. Lactating mothers also have high concentration of oxytocin. And oxytocin is also known as the love hormone. Love hormone in a way that this kind of hormone is of high concentration, especially for those uh, people or individual who have this feeling of uh, who are in love. Okay, in love with their mother. It can be all forms of love. They have high concentration of oxytocin. Vasopressin is another peptide hormone. This is an antidiuretic hormone. It targets the kidneys and helps to limit urine production to keep body fluids up during dehydration. So this is the antidiuretic hormone. So when we say antidiuretic hormone, this is the opposite of diuretic. So there are diuretic compounds uh, that are included in our diet, for example, or drinks. The diuretic substances like coffee, chocolates, everything that contains caffeine are diuretic compound. When we say diuretic, they catalyzes the loss of water and excreted via the urine. So remember that our goal is to maintain the concentration of water in our system for us to look healthy, to look young. And we need to drink more than 8 glasses of water a day, especially in during the hot season. Now, This diuretic compounds catalyzes the loss of water. Now, what is vasopressin? They are the antidiuretic hormone. They prevent the loss of water during dehydration. So, they target the kidneys and helps to limit urine production to keep body fluids up during dehydration. So instead of being dehydrated, our cells to be dehydrated, what vasopressin do in times of uh, we cannot replenish the loss of water. For example, in a day, all you are drinking are diuretic substances. You drink soda, you drink coffee, you drink milk tea. Uh, you are even drinking the in terms of size, uh, the biggest or the tallest. Okay, and then you are only sipping water, okay, a small cup of water in place of this. So remember, every time that you will be drinking diuretic substances, you should also replenish this with an equal amount of water. And there are times that we cannot replenish the same amount of water, and as a result, this diuretic substances catalyze the loss of water in our system. Now, what if in a day you really have uh, a very few amount of water intake? What vasopressin do is they limit the urine production. Okay? And that's the time that you observe that you are producing a yellow orange or a dark color urine, which means that this is already high concentration of solute. There is already a high concentration of solute being dissolved in your urine. And a very few amount of urine is being excreted. This happens to prevent dehydration of the cells. And this usually occur or this usually result to some disorder like urinary tract infection. Because instead of excreting the wastewater, you are immersing the cell in the water that should be already be excreted. But since this vasopressin can't let go of this water because otherwise your cells will become dehydrated and will even have a more serious disorder, then vasopressin would use this water to prevent dehydration. And this is your antidiuretic hormone. So if you are already experiencing a limited amount of urine excretion and a dark color of urine, that is already a sign that you need to replenish your water intake. So these are your two hormones, oxytocin and vasopressin. These are your peptide hormone. Another peptide hormone of biological importance is your glutathione. This is the most abundant single tripeptide distributed in animals, plants, and bacteria. Tripeptide meaning they only contain 3 amino acids. And the chemical name of your glutathione is glutamyl cysteine glycine. So contains glutamic acid, cysteine, and glycine. So I'm sure you are familiar with glutathione. This glutathione is abundantly synthesized in our system, but is decreasing. The synthesis of your glutathione decreases as we grow older. Okay? And glutathione is one of our uh, immune system booster. They prevent us, they prevent the cell from being oxidized. being oxidized from radiation. Okay, from UV light, from radiation coming from your gadget. So, since we have this glutathione, they contribute to the maintenance of the structural integrity of the cellular protein from oxidation. Okay? So, otherwise, your glutathione will be converted into a free radical. And you learn from your organic chemistry what are the characteristics of a free radical. Once your glutathione is converted to a free radical, they are already capable of attacking a healthy cells and convert your healthy cells into another free radical cell. And one free radical cell can attack another can attack another healthy cell and convert this into a free radical cell. So this glutathione prevents the oxidation of your cellular protein by acting as a free radical scavenger. Okay? So once the cell is exposed to radiation, they are being converted into a cell-free radical. So radiations are not only from the sun. So before, during our time, the only sources of radiations that we have are the UV radiation coming from the sun. When we expose ourselves from 10 a.m. to 2 p.m., there is an intense concentration of radiation that is being released. Uh, Watching TV during our time, we were required to have a distance of at least 1 meter from the television. We cannot go near to the televisions because our parents informed us that it releases radiations. Okay, it releases radiation, so there are harmful radiation, so we are not even allowed to go near the televisions. That was before. Okay, so look at us nowadays. So the radiation coming from the suns are more intense, especially during the ano ba, El Nino or La Nina, during the hot seasons. Okay, wherein the sun is releasing a large amount of radiation coming from the heat being released, and that is also the reason why nowadays babies are being exposed to sunlight for about 15 to 20 minutes only. You can no longer expose the baby uh, to absorb the UV radiation for even an hour. Okay, there is already a limit. So unlike before, okay, so if you want to strengthen the lungs of the baby, you let them you expose them to sunlight between 7 a.m. to 8 a.m. But now it is being controlled around 15 minutes only. Now, because of the vast radiations being released by the sun. And aside from that, all the gadgets that we are using contains radiation. uh, the types of radiation, all your alpha, beta, gamma radiations are being released in every gadget that we are using. And we are exposed to this radiation how many hours a day? So especially for those students and at the same time gamer. Okay? So how many hours of uh, you spent with your gadget exposing your eyes and yourself to this radiation. Okay, so there is an effect. But since you are still young, you have an abundant concentration of glutathione. And the main function of glutathione is it acts as a free radical scavenger. So when we say free radical scavenger, your glutathione removes all the free radical from the oxidation reaction. They remove all the free radical, the cell free radical, all of the healthy cells that are converted into a cell free radical. So they collect all the free radical and recycle the glutathione. So you have your glutathione that picks up the free radical, forming the glutathione free radical. Since your glutathione free radical are capable of forming a disulfide linkage because it contains glutamic acid, cysteine, and glycine. So take note, your cysteine is capable of forming a disulfide linkage. So your cysteine will bond with another cysteine of another glutathione to form the disulfide linkage. And take note that to form this one bond, it requires two electrons. And those electrons are this free radical. When we say free radical, they are the unpaired electron. Okay? So collecting this unpaired electron will form now your GSSG or the oxidized glutathione. Now your oxidized glutathione, GSSG, is being reduced by the enzyme glutathione reductase and recycle your glutathione into 2 moles of glutathione plus the enzyme is being recovered. Now again, your glutathione, can you see my cursor? This one, the GSH, your glutathione will react again with another free radical and form the GS free radical. GS free radical will form a disulfide linkage with another GS free radical to form the oxidized glutathione GSSG. Thus, the protein is not structurally modified, only the glutathione. So, GSSG will be reduced by the enzyme reductase to recover your glutathione. So, this is a cycle. So, for as long as you have enzyme, okay, that synthesizes glutathione, this peptide hormone glutathione, you have a free radical scavenger. For as long as you have the enzyme glutathione reductase, you will always recover this glutathione from the oxidized GSSG. Okay? So, but take note, this enzyme that synthesizes glutathione, this enzyme, glutathione reductase, are already within us since birth, okay? And they are busy working, especially nowadays, say, 24 hours a day, 7 days a week, 365 days a year. As in natutulog na kayo, sila nagtatrabaho pa, picking up the free radical scavenger. Bakit? Natutulog ka na pero yung cellphone mo hawak mo pa rin. Yung cellphone mo nasa tabi mo pa din. Yung cellphone mo nasa dibdib mo, nasa bulsa mo. They are still releasing radiations. Okay? And this is a very harmful this releases harmful radiations. Pero mahirap maniwala dahil hindi nyo nakikita. Okay? You will only have your realization if you already have problems with your eyes and you are already running out of your glutathione. Once you run out of glutathione, disorder will occur. Okay? So, the reduced species GSH contain free sulfidyl group and the oxidized form is GSSG. Contain a disulfide linkage forming an intrachain disulfide bond. Okay, so good because we now have the artificial glutathione. But take note that when you say artificial glutathione, they are already synthesized in the pharmaceutical laboratory. Okay? So, and glutathione are being uh, advertised nowadays. I think the first glutathione that was being artificially or synthetically produced is your MET, yung MET. And I think uh, MET is the first glutathione that was being produced. But who are those people drinking this MET or this glutathione? Okay, usually at the after the age of 30, as I have mentioned, as we grew older, the concentration of glutathione is being depleted or is already decreasing. So especially when you are already on your 40, 50, so glutathione can no longer be synthesized in our system. That is the reason why most of there are some individuals or people who are taking the artificial glutathione because they function as a free radical scavenger. They function as an antioxidant. They prevent the cell from being oxidized, specifically from radiation. So the effect of your glutathione, which was being advertised as a whitening effect, that is just a secondary effect of glutathione. But glutathione is more of uh, immune system booster because they function as antioxidant, specifically from uh, the cell being oxidized by radiation. Okay, but be careful also. Anyway, you are still young. Okay, you don't need glutathione. They are abundantly synthesized in your system. But during our time, that was the result of research. During our time, at the age of 30, almost uh, siguro 1% glutathione na lang were being produced by our system. And during our time, we are seldom exposed to radiation. Okay, uh, the main source of radiations that we had before is only the sunlight and the television. Okay, na black and white. Hindi naman, colored na pala. Okay, but still, uh, during our time, the research is for us people to be able to utilize properly our glutathione, and glutathione are abundantly synthesized up to around 30 years old. But what about you nowadays, wherein you are already consuming your glutathione, you are already exhausting your enzyme 24 hours a day because you are very much exposed to your gadget. So make sure that if possible, okay, if there will be some chances wherein you will be sleeping, make sure that you place them in a table or not within the vicinity of your body. Okay, so and uh, during school hours or when we are having on site, stay away from your gadget, from these sources of radiation. Do not even It is not even advisable to place your cell phone in your pocket, okay, in your hands. Especially to you dentistry students, you should take care of your hands, okay, because they will be the sources of your uh, successful career. Your hands are very precious. So make sure that you do not expose your hands to radiations that much. Okay? Another peptide hormone is your glucagon and insulin. So glucagon and insulin was mentioned to you in the metabolism in the study of carbohydrates. What is glucagon? This is a peptide hormone which has a profound effect on glycogen metabolism. What is glycogen? Remember, any stored glucose or any excess glucose in the bloodstream is being stored in the muscle, in the liver, in the form of glycogen. Okay? So, every time you eat an excessive amount of carbohydrates, your excess glucose are being converted to glycogen. Okay? So, now, what is glucagon? This is a peptide hormone which has a profound effect on glycogen metabolism. Why? Okay. And this is secreted in the pancreas during fasting. During fasting state when blood glucose level are decreasing, especially now that the Holy Week is coming, or for our friends, the Muslim friends, they do Ramadan for 30 days. Okay? And they are not eating, not drinking during daytime for as long as there is a once the sun rise, they will uh, they will uh, wait until the sunset before they can, I think, before they can start eating again. Okay? But during daytime, these are ordinary students, workers, employees that observe this Ramadan, and uh, there are really they are really obedient in doing this without eating anything, but they survive. Okay, they survive for the whole day. They do lecture, they do laboratory, on-site laboratory, for example, and then some students will be having their lunch, merienda, snacks. But for them, they observe this Ramadan, so they do fasting. Now, how they survive? Okay, since we have glycogen stored in the muscle as well as in the liver, our glucagon, our peptide hormone, during this time of fasting, will initiate the enzyme to hydrolyze glycogen and to produce glucose to maintain the normal blood sugar level, which is 70 to 100 milligram of blood glucose for every 100 ml of blood volume. Okay? So it is your glucagon that will initiate the enzyme to withdraw glucose from your stored glycogen. This is also the reason why there are some uh, victims, for example, of earthquake who were being trapped for 1 week or more than 7 days, but they were able to survive, okay, without eating anything because of glucagon, especially if you have a lot of stored glycogen in your muscle. So your glucagon will initiate the enzyme to withdraw glucose from the stored glycogen and produce glucose to maintain the normal blood sugar level. The process of hydrolyzing your glycogen is what we call glycogenolysis. Okay? This is the breakdown of glycogen to alpha D glucose to maintain the normal blood sugar level. Take note that I am emphasizing alpha D glucose because there are two forms of glucose. You cannot merely claim that it is glucose. It should be alpha D glucose because the other glucose, the beta D glucose, is produced from cellulose, and we do not have the enzyme which is responsible for digesting this beta D glucose. Okay? So the synthesis of glycogen from glucose is glycogenesis. Glycogenesis. And the breakdown of glycogen is glycogenolysis. Glucagon also stimulates gluconeogenesis. What is gluconeogenesis? This is the production of glucose from non-carbohydrate substances such as protein, accompanied by increased urea production. Remember, there are people who do not eat carbohydrates, okay? And they are only depending on protein and fats. So they don't eat rice, they don't eat pasta, they don't eat bread because they don't like glucose. Okay? So usually these are the conscious people who are maintaining their figure, uh, because remember, any excess glucose can be stored in the form of glycogen and it will be deposited into your muscle as well as in the liver. So there are people who are really uh, uh, avoiding glucose or carbohydrates and do not include them in their diet. Okay? So what will happen to them? So usually they produce glucose from protein and other biomolecules. So protein or lipids can be also a source of glucose. So specifically from protein. Okay, so because there are amino acids that can be converted to glucose through the process of gluconeogenesis. So this is the production of glucose from non-carbohydrate substances such as protein, and this happens in the liver. But again, if you are using protein as the source of your glucose, there will be an increased production of urea. Okay? There will be an increased production of urea because take note that for every catabolism of protein, for every time you are breaking down your protein, you also have your amino group carbon dioxide which are being converted to urea. So glucagon stimulates glycogenolysis, the breaking down of glycogen, and gluconeogenesis, the breaking down of non-carbohydrate substances that lead to the production of glucose, preventing the development of hypoglycemia. Take note that prolonged hypoglycemia could lead to death. Okay? So the opposite of this is your insulin hormone. This is also a peptide hormone. I'm sure you are familiar with insulin. Insulin is a peptide hormone secreted in the pancreas. It facilitates the utilization of glucose and increases the uptake of blood glucose by muscle and tissues and liver. Okay, it enhances glycogenesis, the formation of glycogen from excess glucose in blood. So, if glucagon prevents hypoglycemia, insulin prevents hyperglycemia. So, any excess glucose in blood is being collected by an enzyme. Now, this enzyme is being initiated by the peptide hormone insulin. You may have the enzyme, but if you do not have this peptide hormone insulin, the enzyme will not work, will not receive any message to collect the excess glucose. Then the excess glucose will stay in the bloodstream and it it if it stay in the bloodstream, therefore the person will experience hyperglycemia. Prolonged hyperglycemia could result to diabetes mellitus. So that is the reason why there are some people, take note that insulin is also a peptide hormone, and this peptide hormone are synthesized via the enzymes with the use of enzymes also. Okay? So that is the reason why the geriatric patients, usually the geriatric at the age of 60, 50, their body can no longer synthesize insulin. Okay? So those are, I think, the acquired diabetes. If they can no longer synthesize insulin, then that's a time that they undergo medication or treatment that um, to 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 uh, to have your insulin work. But I think after doing some medications and after some time, a period of time, if the patient is no longer responding to through medication, oral medication, then they already inject themselves with insulin. Okay, so they already inject themselves with insulin. Although this can also be through a genetic uh, disorder wherein it is being inherited from their parents, and even at a young age their system can no longer synthesize or produce insulin, and at their early age they were already injecting themselves with insulin. So take note that it is important in removing any excess glucose in blood and convert it into glycogen. So that is the reason why insulin enhances glycogenesis, the formation of glycogen from excess glucose in blood. There are also peptide hormone known as the neuropeptides, the enkephalin, which function as a pain reliever. So we have in our system the enkephalins which function as a pain reliever. So uh, these are pentapeptide made in the brain that act as painkiller and sedative by binding to pain receptors. So although there are also addictive drugs like morphine and heroin that also bind to the same pain receptors, thus producing a similar physiological response, though longer lasting. And they take note that they are addictive substances. That is the reason why morphine is being used as a pain reliever. This is being used as a pain reliever, but it should be prescribed and controlled. Okay? Uh, Nowadays, there are some doctors who opted to prescribe this morphine because this is from a natural extract. This is from plants. Okay? So they are not synthetically prepared. They are not synthetically prepared. So they are simply extracted from the 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 plants. So and there are some doctors believe that opted to use this rather than the synthetically prepared pain reliever. But usually these are being utilized or used and prescribed uh, during the terminal cases, terminal stages, or severe. Okay, for those who are already experiencing uh, severe pain due to some terminal illnesses like cancer stage 4. So they are already they are already prescribing this. Although again, this should be controlled because this is addictive. Enkephalins belong to the family of polypeptide called endorphins, which are known for their pain-reducing and mood-enhancing effect. So we are also they are naturally occurring in our system. These pain relievers, okay, the enkephalins. So one of the enkephalin is methenkephalin. It is a pentapeptide made up of tyrosine, glycine, phenylalanine, and methionine. And the other one is leucine enkephalin. This is tyrosine, glycine, phenylalanine, leucine. Okay, so this is the other the other main enkephalin. Okay. So if you will be hydrolyzing your protein molecule, take note that your protein molecule usually comprises of more than 40 amino acids joined together. So from protein to a bigger molecule and then down to peptides. Okay, so peptides can be decapeptide, nonapeptide, pentapeptide, tripeptide, down to uh, amino acid, alpha amino acids. Now, what about the protein? The proteins can be described through their structures. And we have the so-called primary structures, secondary structures, tertiary structures, and the quaternary structures of protein. The primary structure of protein is the simplest, okay? This is a simple sequence of amino acid joined together by peptide bond. So all bond angles are 120 degrees, giving the protein molecule a zigzag arrangement. Here, this is just a continuous bond of your amino acid, but the difference here is that the alkyl group are being positioned in an alternate way. Below the plane, above the plane, below the plane, above the plane, below the plane, above the plane, below, above, the alkyl group. Okay? Resulting to a zigzag arrangement of your primary protein. So this is the simplest protein structure, your primary protein. The second type is the secondary protein. The secondary structure is the 3D arrangement of your localized regions of a protein. These regions arise due to the formation of H bond between the amino group and the carboxyl group of another amino acid. Two stable arrangement result from this secondary structure. You have the alpha helix and the beta pleated sheet. So most proteins have regions of alpha helix and beta pleated sheet and the other regions that are random in arrangement. This is the secondary structure of protein. This is the alpha helix. They are helical in structure. And the helical structure of protein results from the formation of hydrogen bond between the amino group and the carboxyl group of your amino acid. Okay? So if there are adjacent carboxylic group and amino group, they form H bond. So as a result, they form a helical structure of amino acids. This is the beta pleated sheet. A beta pleated sheet is a pile of of protein structures or long chain of amino acids, which results again from the attraction between the amino group and the carboxyl group that forms the peptide bond. So take note that the intermolecular forces present in your secondary structure is hydrogen bond. So the cost of the bending of your amino acids is due to the presence of the hydrogen bond between the amino group and the carboxyl group of another amino acid. So because of this hydrogen bond, alpha helix and beta pleated sheet structure results from the secondary structure of your protein. What about the tertiary and quaternary structure of protein? The tertiary structure is the 3D shape adopted by the entire peptide chain. So to maximize H bonding with water, the nonpolar side chain are stabilized by London dispersion forces in the interior structure. So that means that your protein molecules, okay, the intermolecular forces of attraction present in your protein molecule with tertiary and quaternary structures are H bond and London dispersion forces, or the van der Waals forces of attraction, or merely van der Waals forces of attraction. Okay, these are both intermolecular forces of attraction. The London forces, the London dispersion forces occur in all organic compounds, especially the hydrocarbon chain. Okay, so if your amino acids are being held by, or if your amino acid is with an alkyl group of hydrocarbon, say valine, leucine, isoleucine, they are being, uh, they are attracted to another alkyl group, which are also made up of hydrocarbon, like again valine, glycine, leucine, isoleucine, and they form the attraction between these hydrocarbon groups are known as the London dispersion forces, or the van der Waals forces of attraction. This is the weakest, okay, although there is an attraction. Another intermolecular forces of attraction that is present in tertiary and quaternary structure is the uh, electrostatic interaction. These electrostatic interactions are present in amino acid with charged side chain, like the acidic amino acid, basic amino acid. They have charged side chain that results to electrostatic interaction with another amino acid. So H bond, London dispersion forces, electrostatic interactions are the intermolecular forces of attraction present in a tertiary and quaternary structure of protein. There is another, you have the disulfide bond. So if there is two cysteine within the vicinity or at the opposite side of the chain, they form the disulfide linkage. So there are a lot of IMF present in your protein molecule, specifically on the tertiary and quaternary structure. So what does it mean? That your protein becomes more stable. Okay? They increases this IMF results to high boiling point, high melting point, and oftentimes results to a bigger molecule, more compact molecule that makes them insoluble in water. So this is a type of disulfide bond that result from a tertiary or quaternary structures of protein. So it can be intramolecular. When we say intramolecular disulfide bond, that means adjacent cysteine. You have an adjacent cysteine. Cysteine, cysteine. Therefore, they are capable of forming a disulfide bond. Okay? Another is the intermolecular disulfide bond. So when you say intermolecular disulfide bond, these are from two different peptide chain, okay, which are on the opposite side. So this is the first peptide chain. This is the second peptide chain. So on the opposite side, they can also form a disulfide linkage. So when your cell is being oxidized, it can be prevented by forming the disulfide linkage so that your protein molecule will not have an immediate disruption of the structure. Okay, that is the one of the main function of your uh, cysteine. They prevent the cellular structure of your protein molecule because that is important, especially if the protein is an enzyme. Okay, if there are some oxidizing agent, your enzyme can be affected by this oxidizing agent because they are also amino acid, they are also protein, they are also made up of this amino acid. But if there is cysteine within the chain of your protein molecule, they can prevent from being oxidized by forming the disulfide linkage. And you can regenerate this peptide chain by having the glutathione reductase. This is an example of a tertiary and quaternary structure of protein. Okay, so we also have here the helical structure, which is found in the secondary protein. And in the secondary protein, you have the helical structure. Take note that results the this results because of the presence of the hydrogen bond. Okay. We also have here examples of a disulfide bond from the uh, opposite side of your peptide chain. And then you also have the London dispersion forces from phenylalanine and uh, leucine. And you also have H bond that occurs from serine and uh, another amino acid with OH group is this threonine. And then you also have your electrostatic attraction between the acidic amino acid. I think this is glutamic and this is arginine. Okay? Arginine, your amino group. I'm sorry. This is lysine. Okay? So the amino group and the carboxyl group forms the electrostatic forces of attraction. So again, you still have your H bond, H bond, H bond. That results to the coiling and folding of your amino acid that results to your quaternary and tertiary structures of protein. The quaternary structure of the protein is the shape adopted when two or more folded polypeptide chains come together in one complex protein molecule. So insulin consists of two separate polypeptide chains linked by intermolecular disulfide bond. You also have your hemoglobin, which are consists of four subunits of uh, tertiary structure held together by intermolecular forces into a compact 3D shape. So this is your primary structure, uh, and then you have the beta pleated sheet and the alpha helix. This is the 3D structure of your tertiary structure of protein because you have more intermolecular forces of attraction present in your 3D, in your tertiary structure. You have your H bond, you have your disulfide linkage, you have your electrostatic charge of attraction, and you also have your um, IMF are the London forces, H bond, disulfide, and the uh, electrostatic charge of attraction. So there are four IMF present in your tertiary structure of protein that results to folding of your amino acid. Okay? So this is the resulting structure of your 3D structure of a tertiary structures of protein. Now, this tertiary structure of protein once bind to another tertiary structure of protein, you form now the quaternary structure of protein. When two or more subunits of the tertiary structures are being joined together, you form the quaternary structure of protein. And an example of that is your hemoglobin. There are four subunits of tertiary structure, and they are being held by the iron, okay? By iron, forming your hemoglobin. Proteins are generally classified according to their 3D shape. So as a result, they end up with either a fibrous protein, which are composed of long linear polypeptide chain that are bundled together to form rods or sheet. Fibrous protein are insoluble in water and serve as structural roles like your collagen, uh, fibrin, keratin. They are usually fibrous protein. Globular protein are coiled into compact shapes that are water soluble, like your enzymes, and transport protein, which are globular. So keratin are proteins found in hair, hooves, nails, skin, and wool. They are made up of two mainly alpha helix chain coiled around each other in a superhelix. These coils wind around other coils, making larger and stronger structures like the hair. Collagen requires three chains in a superhelix. And again, this is just a reminder that vitamin C helps stabilize the collagen chain. And when missing, they poorly form collagen fibers. As a result, uh, it results to the sagging of skin. So when there is already sagging of the skin, especially in the face, that means that is already a sign that you lack vitamin C because your collagen cannot be formed and synthesized. And remember that collagen is important in maintaining the the stiffness of your protein in the skin. So this is the fibrous protein. This is a uh, alpha keratin made up of alpha helix, and a supercoil formed from two helices form the strands of hair. You also have your collagen, which is a triple helix. Example of globular protein. Hemoglobin and myoglobin are globular protein and conjugated protein, meaning when you say conjugated protein, they contain both protein and non-protein component. Okay? Can be glycoprotein, meaning a small amount of carbohydrates in your protein. Can be lipoprotein. You have protein, a bigger molecule, but with a small amount of lipids. So you look at the surname, lipoprotein. The surname is protein. That means it is really protein with a small amount of lipids. Glycoprotein. It is really protein with a small amount of carbohydrates. But if it is already proteoglycan, so glycan are carbohydrates. So it is carbohydrate with a small amount of protein. So their non-protein unit is a heme, an organic complex surrounded by iron. The ferrous ion binds the oxygen gas in the bloodstream. Then the hemoglobin protein transport the oxygen to wherever it is needed in the body. Or if needed, the myoglobin stores oxygen in tissues. So this is the heme unit, wherein you have your heme, the protein molecule, and you have the iron at the center. Myoglobin has 153 amino acids in one polypeptide chain, and you have the heme, wherein you have iron at the center. And this is your hemoglobin. There are 4 polypeptide chain in each carrying a heme unit. So you have your protein with iron at the center. Hemoglobin and myoglobin. So carbon monoxide is poisonous because it binds 200 times more strongly to iron than does oxygen. So hemoglobin complex with carbon monoxide can no longer carry oxygen and cells will die from lack of oxygen. The sickle cell anemia is a disease where a single amino acid is different in two of the subunits of hemoglobin. So the red blood cells containing these mutated hemoglobin units become elongated and crescent, known as the sickle shape. These red blood cells will rupture capillaries, causing pain and inflammation, leading to organ damage and eventually a painful death. Okay, this is a replacement of the amino acid uh, due to the exposure of your uh, exposure of your hemoglobin to some mutagen. Okay, so exposing your hemoglobin, your cell to mutagen results to the replacement of this amino acid that that destroy the structure of the normal hemoglobin structure, and it results to an elongated and crescent shape. Okay, so usually this happen when uh, there are some antibiotics that can cause this the formation of sickle cell anemia, and most of these antibiotics were already banned uh, by the Bureau of Food and Drug because they affect the formation of this. Uh, they affect the hemoglobin and causes to produces the sickle cell. Okay, prolong formation of this sickle cell can lead to uh, leukemia. The hydrolysis of protein and protein denaturation will be discussed in the laboratory. We have an experiment that will deal with protein hydrolysis and denaturation. Although when we say protein hydrolysis, that is just the same with carbohydrate hydrolysis. Remember, we hydrolyze starch in the laboratory through the stages of from starch to dextrine to maltose and alpha D glucose. So same with protein. We will be hydrolyzing protein from protein to polypeptide, peptide, and then amino acids. So breaking this protein molecule forms your peptide. Breaking your protein molecule involves breaking the peptide bond. Okay, by addition of acid, bases, or enzymes. So they can also be hydrolyzed with the use of this. So in the body, the enzyme pepsin is found in the gastric juice. They are found in the stomach, and they are responsible for breaking your peptide or protein molecule in the stomach, because pepsin is responsible for the digestion of your protein. And pepsin is found in the stomach. In the intestine, specifically in the small intestine, enzyme trypsin, chymotrypsin further hydrolyzes your protein molecule and convert this into a free amino acid or individual amino acid. We'll have the denaturation on the laboratory. And then the next topic will be enzyme by Thursday, which are also protein molecule. Do you have any question, class? How are you? Tomorrow is Wednesday. We will be having an online classes uh, for the laboratory. Okay? So for the whole week, I think we will be having an online classes. So lipids will be online. We will discuss this virtually. Same with what is the next? I think protein. So probably by Friday we will be discussing the denaturation of protein. How are you? Yeah, next week we will be having an on-site after the jeepney strike. I think our the CEU, the vice president of the CEU, uh, have the opted to have the online classes for the whole week. Regarding class sa mga check-in activities, pag walang due date, minsan tuloy nalilito ako sa inyo. Pag walang due date, if there are no due dates indicated, usually dinidelete ko yan. Okay? Because we receive our modules and we edit modules. And bago ko i-publish yan, chine-check ko muna, dinidelete ko muna yung mga hindi nyo gagawin. But there are times siguro na hindi kayo naka-refresh ng gadget. Kasi I cannot see those your questions like a quiz in amino acids. Saan ba yun? Bakit wala sa akin? If I will be showing you my module, ito lang ang naka-assign sa inyo. The all quizzes which are conducted in Canvas, and if you have some concerns, you just inform me, you just remind me because I will be rechecking it. And somebody already reminded me uh, regarding module 4 pretas. Okay, eto lang ang nasa akin class. For module 4 proteins. What about module 4 check-in activity? Your module 4 check-in activity is about peptides. So pretas, and then you have the module 4 check-in activity. This is already peptides. This is also peptides. And then we will be having a module 4 post-task. This is a quiz. Okay? But I haven't published it yet. So I will schedule this after the discussion of protein. Okay? If I will be opening this module for check-in activity 3, this is show the formation of the following peptides. Ayan ang check-in activity 3 nyo. Show the formation of the following peptide. And then, back. Ang module 4 check-in activity, the other check-in activity. Check-in activity 4. Explain using two sentences the biological significance of the following peptides: glucagon, insulin, ceruloplasmin, and oxytocin. There are only 2 check-in activities that I have assigned to you. Activity 1. I did not assign check-in activity 1 and activity 2. So if you observe, kaya nga always refresh your gadget class. I only assigned pre-task, this one, pre-task, and then I have here module 4 check-in activity 3 and module 4 check-in activity 4. Okay? And then you will be having the module 4 post-task. No activity 1. There is no module 4. Module 4 check-in activity. Okay. Only module 4 check-in activity 3 and 4. I do not know why it reflects in your canvas. May deadline po kasi siya lumabas po sa dashboard namin. Lecture ba yan, Angel? Saan kaya? Ba't sa akin wala? Wala naman sa akin. Wala po sa mismong page ng module. Nasaan? Assignment page. Teka lang ha. Baka may naka-ano dito. Module 4. Module 4, pre-task. Module 4, post-task. Quiz on lipids. Wala rin sa akin. Tignan ninyo. Module 1. Assignment to ha. Under ng assignment. Module 2, Module 2, Module 3, Module 4. Check-in activity 3, due date March 13. Ito yung peptides nyo. Module 1, post-task. Module 1, pre-task. Post-task, pre-task. Module 4, post-task. Quiz on lipids. Module 6. Module 8, Module 1. Wala. Module 4, check-in activity 4. Ito yung ito yung peptide hormones. Bioenergetics. Ito siguro, naka-check. Unpublished ko na. Pero wala namang date. Walang date. Ito ba? Module 4, check-in activity. Quiz on essential amino acids. Walang date to. Walang due date. Nakikita nyo? Activity 1. Sige, back pa tayo. Module 4, activity 2. Module 4, Module 4, check-in activity 3. Module 4, post-task, pre-task, 51996728884. Check. Ito nga. March 6, patingin. Quiz on amino acids. March 6, Feb 25. Anong itsura niya? No answer. Okay, next. No question, no answer. Kayo naman. Anong problema nyo dyan? Wala palang tanong. Gusto nyo lagyan ko? Gusto nyo ba siya talaga? Kaya nga. Okay. So, sige, lalagyan ko ng tanong. I will put questions. Walang tanong, walang choices, walang sagot. Di wala. Submit quiz. Because this is probably for editing. So, cancel na natin to. Delete. Yan. Hindi nyo na makikita yan. Unpublish. Do not answer that. Number 1, there is no question for you to answer. Okay? So, edit. I think Yasan already asked this, and I told Yasan to ignore and disregard this quiz. But I do not know why you still insist. Do you really want to have another set of quiz for this? I have already informed you to disregard. Kasi nga walang question. Gusto nyo ba lagyan ko? Ang kulit kasi, sabi ko na nga disregard, sasabihin pa ni Lendy, ma'am, paano po yung ano nga yung follow-up ni Lendy na question? Paano po kung nagsagot na? Paano mo sasagutan? Eh, wala ngang tanong. Diba? Paano mo sasagutan walang tanong? Sige nga. Paano nyo yun nasagot? Believe naman ako sa inyo kung nasagutan nyo yun ng walang tanong, paano nangyari yun? How can you answer questions that wala namang tanong ay siya? Sinabi na nga i-disregard, sasabihin pa sa akin, eh paano po yung mga nakapagsagot na ano kayang sinagot nila? Parang pag ganun ng tanong nyo sa akin, eh ma'am kasi gawan mo na ng tanong para may sinasagot kami. Parang ganun ang gusto nyong mangyari. Pero yung isa kong tanong, hindi nyo pa sinasagot. Ano nangyari sa ating mga dancers? Walang nangyari. Walang nanalo sa inyo. Ang aking Mr. and Miss, meron? Oh, second placer din kayo? Akala ko yung kabilang section ko, second place. Seryoso, Tay? Sige. Oy, grabe ka. Pilapil plus 25 ka dyan. Plus 2. Plus 5 lang. Sa final exam. You give me the list of those who participated. Maski naman yung aking Mr. and Miss Dent, bibigyan ko yan. Meron ba kayong representative? Wala. Meron, di ba? Second year. Nasa section nyo, di ba? Yung Miss representative ng second year. Oo nga, mami. Oo. Nasa inyo din? Kayo-kayo lang din? Ang makeup artist? Sige, Lendy. Facilitate mo yung list. Okay? Dancers, everybody who participated. I will just give you an incentive for the final exam. Anong props? Pati yung props, bibigyan ko ng ano? Anong props? Mga balloons, ganon? Seryoso, pati si Yasan tumulong? Okay. You just remind me. Okay? Because I might also forget it. So during the final exam, for the lecture, you just remind me for the incentive plus 5. Okay? So kung ba gagamitin? Sa Lex, sa lab. O tsaka na natin pag-usapan. Anyway, it's already 8:30. I'll see you on Thursday class. Tomorrow, I'll see you in the laboratory, 9 a.m. Do you have any more concern? I think I have already posted your scores for the lecture, but for the lab, not yet. I was supposed to check your Scantron paper yesterday, but because of we opted to have the online classes, then probably I will just release your score next week during the on-site class. Okay? So if you don't have any more questions, you may go. I'll see you tomorrow. Goodbye, class.
Goodbye, Doc.
Bye. Meron na kayong protein na dash na ano na module for pretas po. Oo, oo, Patrick. Oo. I will review your scores before I will encode it to my e-record. Nakalimutan ko lang i-edit yung instruction. Nakalagay no space pero minalian niya. Oo, sige. Okay. Landy, may concern ka pa?
Wala naman na, ma'am.
Okay. Class, I will end the Google Meet. I will put down the phone.